Thromboembolic diseases are a leading cause of human disability and death, and they are caused by blood coagulation inside blood vessels, which impedes the flow of blood to the heart. In order for blood coagulation to occur properly, clotting factors are required to be present in the blood, and these clotting factors are produced through a process which requires multiple enzymes. One of the enzymes required for the formation of clotting factors is the Vitamin K Epoxide Reductase enzyme, abbreviated as VKOR, which can be seen in this movie as the white-colored structure. This enzyme is important because it produces the reduced form of vitamin K, called vitamin K hydroquinone, which is required by a separate enzyme in order to carboxylate precursor clotting factors, so that they become active clotting factors. In this movie, the VKOR enzyme shown is not identical to the VKOR enzyme found in the human body, due to the fact that the enzyme in this movie has two modifications. The first modification is an amino acid substitution at position 185, where a cysteine residue is substituted for a serine residue. This modification has not been characterized in the VKOR structure being observed in the movie. However, it can be interpreted to be present between the white tube present at the bottom left and the white tube present at the top central right. The second modification is the addition of a 1R, 2R, 1-amino, 2-hydroxypropyl, 4,4-hydroxybenzylidine, 5-oxo-4,5-dihydro, 1H-imidazole, 1YL acetic acid molecule, termed SCRO by the Protein Data Bank, which in the movie can be observed as the structure which is brown in color. This modification acts as an L-peptide linker. The functional action of the VKOR enzyme can be inhibited by the drug Warfarin, which, in the movie, is the small molecule that is lime in color. Warfarin inhibits the VKOR enzyme by binding to the active site of the VKOR enzyme, which is a bundle produced by 4 transmembrane helices. This active site can be seen as the 4 white spiral structures in which the lime warfarin molecule is present. Inside the active site, the warfarin molecule forms 2 hydrogen bonds, one with the second transmembrane helix and one with the 4th transmembrane helix. In the former, a hydrogen bond forms between the double-bonded oxygen atom present on the 22nd carbon atom in the cyclohexane ring of the warfarin molecule and one of the hydrogen atoms present on the amino group of the R group of the asparagine residue that is in turn present at position 222 of the VKOR enzyme. In the latter, a hydrogen bond forms either between the oxygen or the hydrogen of the hydroxyl group present on the 13th carbon atom of the cyclohexane ring of the warfarin molecule, and either the oxygen or hydrogen present on the hydroxyl group of the benzene ring in the R group of the tyrosine residue that is in turn present at position 281 of the VKOR enzyme. Once the warfarin molecule is bound to the VKOR enzyme, this causes a change in the intracellular redox state of the VKOR enzyme, which inactivates the VKOR enzyme and does not allow it to act on the vitamin K to form vitamin K hydroquinone. Due to this reason, the enzyme which carboxylates the precursor clotting factors so that they become active clotting factors will no longer be able to work, as it will not have any vitamin K hydroquinone to use. Therefore, no active clotting factors form, which in turn leads to the blood not being able to clot inside blood vessels. Hence, thromboembolic diseases will be prevented as the blood is not able to clot, showing how important warfarin is in preventing these conditions.